Good morning, everyone. My name is Hiran Vigad. I work as a sales product specialist for LemnaTec Limited. LemnaTec is a UK and Irish distributor of various analytical instrumentation and scientific equipment. We've been working with LemnaTec for about 2 years or so. So I'm very fortunate to have Marcus Jansen here. He's one of the chief scientist biologists from LemnaTec, and we're very much looking forward to listening to your talk, Marcus. But let me pass the mic on to Marcus, who will introduce himself and the company, and we'll look forward to listening to your presentation. Thank you.
Today it's about revolutionizing the physiological phenotyping with the power of spectral imaging. The webinar today is, as said, about spectral imaging in phenotyping, and I'm starting from the basics. So that means I will start with very fundamental technical things, but at the end I will also have some application cases. And of course, I'm looking forward to get some feedback later on, depending on how much you are familiar with phenotyping and spectral imaging. You might have a different point of view to that, but I think it's a good idea for such a webinar to start from the basics. So the company LemnaTec was founded in 1998, and it is a company for digital phenotyping. It has phenotyping equipment that consists of camera systems, that consists of some accessory hardware and software for data analysis, data interpretation, and in the end, of course, value generation. The technologies are suitable for laboratory equipment, for laboratory applications. There is also some equipment for measurements in greenhouses or in outdoor environments. We have customers throughout the world, not only in Germany, not only in Europe, but also several customers in North America, a couple of customers throughout Asia and also Australia. Of course, with technical equipment, locations like Africa or South America always are a bit lagging behind, but we hope, of course, that we can also approach those regions because we see that there is a demand for technical solutions also outside the classical industrialized countries. LemnaTec is a part of the Nynomic Group since August 2019. The Nynomic Group is a holding company that has 10 companies in the group, and all these companies are somehow related to imaging, sensing, and the related technologies and software parts. It is about non-contact, non-destructive optical measurements of different things. We are here on the level of plants and seeds, but there are also companies that work on technical items, that work on wood or food or environmental sensing. So there is a lot of different technology in the group. But let me now start with classical versus spectral phenotyping, an introduction, what it is about today and how the technology is related to the biological applications. In classical phenotyping, people started working with normal photographs in white light. But the question is now, why using spectral cameras? First, let me introduce a bit the classical situation. We have a light source, and there is this normal white light that you use for photographing also in personal environments. And of course, you know from physics that the white light is composed out of a range of wavelengths. And light is then reflected from the sample, in this case from the plant, and it is captured with an RGB camera. And inside the camera, there is a color image generated and stored. And when you want to analyze that, you use image processing software. And of course, you still can get information about the color channels, for instance, the R, G, and B channel in when you do some software processing of a photograph. The step to spectral imaging then is that you, from the beginning on, separate the different wavelengths and not wait until you have to do that in the software. That is one of the key points that I will address here in the webinar later on. So what you can do from a biologist perspective, you might be interested in the morphology and the dimensions of the plant. So how tall and wide and what is the area of the plant. If you have multiple objects like those seeds here, you might be interested in counting them, but also in the same time in measuring. Are they all of the same size, or are they bigger and smaller, and so on? You get the colors as said as a bulk RGB information, but of course in image processing you can look for green areas, that is classically assumed to be the normal status of a plant leaf, but there are also brown areas that come from stress and diseases, and so you can do some assessments of stress and diseases in plants. And of course, applications are not limited to that. There is much more to do in phenotyping. So when we move from classical white light phenotyping to spectral phenotyping to spectral imaging, we have light sources that emit light in different wavelengths. So here is a light source depicted, and it can go in different ranges of wavelengths. The light is emitted in different ranges of the wavelengths. And of course, all these wavelengths are irradiated to the sample, and parts of the light is reflected from the sample. And in this case, there is not an RGB camera, but it is a grayscale camera that looks at the light intensity that comes back from the sample. And in the end, you get a stack of images, depending on how many wavelengths you have used for illuminating, that each of them reflects to a certain, or corresponds to the reflection in a certain wavelength. So you have the color related, the wavelengths related information directly from the camera, and not only in the end when you work in software level. So that of course makes it much easier to do calculations, because you have the information already separated, and you don't need to separate it on software level beforehand. Of course, you can do very similar things like counting, looking at morphology and dimensions. And as said, you have the colors as single channels. But you have much more possibilities in physiology. When it is about stress and disease presence and symptoms, you can do more assessments compared to just seeing there is a color change or something like that. You probably can also see pre-symptomatic changes, and you can calculate indices, vegetation indices, that relate to certain physiological status of the plants. When we talk about spectral phenotyping, it can be multispectral or hyperspectral phenotyping. The easier thing is the multispectral phenotyping, therefore I'm starting with this, and we move on to the hyperspectral phenotyping later on. In the multispectral setup that I'm presenting, as said, we have cameras that record the light intensity reflected from the sample, and we have lights that emit different wavelengths, and it looks like that for a human eye. It looks quite colorful. When we switch through the light, through the wavelengths, then we have different light qualities that are illuminating the plant, and for each of these light situations, we record a photograph or a series of photographs from the plant. You see here cameras looking horizontally and vertically on the plant, and what you don't see, but it is here below the plant, is a turning unit, so the plant can circulate once around, and the horizontal camera takes a series of images during the turning process, so you have a series of views for the plant from different sides. If you need it, And of course, you can do that for all of the different colors, for all of the different wavelengths that we are able to switch on in that cabinet. So when we have, for instance, red light from the light sources, from the LEDs coming onto the plants, it is known from biology that chlorophyll absorbs red light. And we are here with a wavelength that is corresponding to the chlorophyll absorption. So only a little of the light is reflected and reaches the camera. And in the end, on the photograph that the camera is recording, the plant looks quite dark because the red light is absorbed from the healthy plant. It would be different when the plant is stressed or diseased or even dead. But in this case, the plant is absorbing the light because the chlorophyll is using that for photosynthesis. When we switch from red to far red, which is only a little different in the wavelength range, in the spectrum, then the plant is reflecting much more of that light, and the resulting image looks bright. So the far red light is more or less completely reflected, and the plant looks bright. And now you have the situation that there is an absorption and a reflection for the same sample material, and you can start calculating with these different responses, for instance, to make a so-called vegetation index. Technically, we use that range between 380 and 850 nanometers out of the light spectrum, so that's more or less covering the whole visible spectrum. There is a LED that is close to the border to UV, and there is an LED that is just in the near infrared range, and all the others, 6 other LEDs, are in the classical visible light range, where what you also can see as a human when you look at samples illuminated with white light. And when we illuminate the plants or the samples with all these different wavelengths and we switch, there is this one case in this example where the red, which is inside the chlorophyll absorption, then is looking dark, whereas the others are looking bright. And of course, when we have different types of samples, this pattern of reflection and intensity can change. And we have here an example of the channel-wise imaging and deriving some information with 3 leaves, one of which is fresh. The second is more or less completely dried out. It's more or less dead. And there's a 3rd leaf that has some spots here, and these spots result from a nutrient deficiency in that plant. So that is a partially damaged leaf. When we now look at the different channels, and to make it a bit simple, we have not displayed all of the channels here, but the key channels, first red, green, and blue, so the classical channels that make up a color image. And you can also calculate such a classical color image out of the channels, but you see here already there are differences how the leaves reflect or absorb the different channels of the spectrum. The leaf that has almost lost all its chlorophyll is reflecting very strong in the red. It is also reflecting in green and in blue. The leaf that is intact not only has chlorophyll, it has also some other leaf pigments like carotenoids inside. It is reflecting a little of the red, of the green, and the blue channels. So it is absorbing with the pigments parts of the light, but it is also reflecting other parts. The thing here in the damaged leaf, you see within one leaf that there are differences. By the way, what you see here as an output is always a color-coded image from the computer. It is not what the camera directly sees, because what the camera directly sees are these grayscale intensities, but we as a human cannot really work well with grayscale intensities. So there might be differences between this and that, and we just don't see them because the human eye is not trained to work on differences between grayscales. Therefore, the computer output is enhancing that with a color coding so that we see better where are the differences. An application example, as I said, is a vegetation index like the NDVI. The NDVI is a ratio out of the difference between the reflectance in far red and red, and I have been showing that an intact plant is absorbing red but reflecting far red, and that's the difference between far red and red, and it is divided by the sum of the reflectances from far red and red, and that's the normalized difference vegetation index, NDVI. How it works is nicely to see here. In far red, the whole vegetation, the whole leaves, regardless whether they are intact or not, are reflecting. That's coming from the outer structures, that is the cell walls and surfaces. It has nothing to do with the chlorophyll. In the red channel, the chlorophyll is absorbing. The intact leaf is looking dark, much darker compared to the dead leaf, which has almost no more chlorophyll. So in the end, in the NDVI, the dry leaf, the dead leaf, is very dark. It's close to 0, while the intact leaf is bright, so close to 1. And you see already some differences within the intact leaf. There is this midrib, this vasculature, indicating that there is less chlorophyll in the vasculature of the leaf, while the leaf blade has much more chlorophyll inside. And when you now look at the damaged leaf with the nutrient deficiency, you see here these spots where the leaf turned brown, but also some surrounding of that brownish area has a lower ratio of NDVI, because there the chlorophyll has been degraded, and therefore the NDVI decreased. So this is a simple but typical application example, and when you now would apply that to your samples for instance, in a stress assessment, in a breeding program, or in a plant pathological assessment, then you would be able to use that technology to detect stress impact or to detect pathogen colonization. We can do even more with that technology when we put some filters in front of the camera. So far, we only had the light intensity at the grayscale, but here we can also filter out some wavelengths when we only want to see part of the spectrum and not the whole light intensity that comes back from the sample. In this case, we have a bandpass filter at 695 nanometers. That is the emission maximum of chlorophyll fluorescence. So we can see fluorescence that comes from chlorophyll, and we can excite that fluorescence when we send blue light onto the plants, because you see here not only in the red part of the spectrum, but also in the blue part of the spectrum, there is absorption by chlorophyll. And when the chlorophyll is absorbing light, it takes up the energy. First, it has some heat loss to a certain level, but from this lower excited state, then it gives away the energy partly as fluorescence, and that this is what we can image here with the filter in front of the camera. When we excite with low wavelengths, so close to the UV, we see already some fluorescence when we excite with blue light, which is pretty much inside the absorption maximum to chlorophyll. We see a lot of fluorescence, and when we go a bit beyond in the blue range, we see that the fluorescence becomes lower, but we still can differentiate between healthy and damaged tissue. So it is not only meaningful to look here inside the, let's say, so-called optimum, but it's also meaningful to look a bit before and beyond to be better able to differentiate between properties of the sample. For instance, here in a stress situation, it might be important or it might be meaningful to compare these samples at different excitation wavelengths and compare how the output is depending on the excitation. What we also see is that the leaf, which looks pretty much dry and dead, still has a little fluorescence here at the basis. That means there might be some remaining chlorophyll inside that we don't see by eye, but we can make something visible which is not visible to humans, and therefore we can dissect properties of the samples better when we have spectral and fluorescence information compared to a situation where we only have a photograph. We can also have a look at fluorescent biomarkers, for instance, at GFP. Take a different filter, of course, and we take a different excitation compared to when we look at chlorophyll. And you know, GFP is not only one protein, there are several proteins available. Some have more difference between the absorption and the fluorescence emission maxima here in the spectrum. In other cases, they are more closer together. So of course, when they are a bit more separated, then it's easier to image. When they are closer together, it's becoming harder because in the image, you see also some reflection of the excitation light. But you, of course, then have a sum of the background illumination and the fluorescence emission that comes from the GFP. So therefore, it's always important to take proper controls, because without a control, you would never know, is it a signal that comes from the excitation light, which cannot be completely filtered out because of that overlap, or whether this is a signal that comes from the fluorescence where you are looking for. So one of the messages, which is key for more or less all imaging and image processing applications, is work with a control where you check the target trait versus a situation where the target trait is absent, and then look at the ratio between the signal and the background. Moving on from this multispectral situation to hyperspectral phenotyping. Multispectral, as said, there is a series of different images with different lights, probably also with filters when you are looking at fluorescence, but it is normally a distinct and quite limited set of images that you are analyzing. The situation is different in hyperspectral phenotyping because then you have a scanning sensor that has X and Y as the area of the sample, and in that direction it is a stack of many images that correspond to a broad range of wavelengths. So you would not have a few separate wavelengths like these 8 wavelengths in the multispectral imaging, but a data cube where each pixel corresponds to a spectrum that consists of many data points. So you have much more data. Much more data also means you need more storage, you need more processing capabilities to work with the data. The measurement principle is similar, but somehow different. In the measurement, you cannot work with LEDs, but you need a halogen light as light source. again, this halogen light is composed out of different wavelengths. They are irradiated on the sample of the plant. And then you have a hyperspectral scanning line camera that is now scanning the reflected light by the different wavelengths. And of course, you again have images that correspond to different wavelengths, but not only that two that you see here in the example, but as you could see here in the previous image, a big stack of images as a data cube. So you get not distinct points for wavelengths, but you get a spectrum. Here you also see absorption by leaf pigments and scattering, for instance, here by the cellulose air boundaries, which is in the near infrared, so the plant surfaces. And this is the so-called red edge. That's also the reason why we have these red and far red imaging in the multispectral. You want to capture that point where is the transition between absorbing by chlorophyll and reflecting by the outer surface. Of course, you get it much more continuous when you have a scanning hyperspectral camera compared to having a multispectral camera that, for instance, has one point here, one there, and a third over there. When we run hyperspectral cameras on plant samples, we can, of course, again work with vegetation indices. And here you see an example where the NDVI was recorded at large scale. And you see a really big phenotyping system, which is located at Rothamsted Research, which is a bit north of London. It is 10 meter wide, and I think it's about 100 meter, no, 100 meter, probably. It's quite long, of course. It's moving on rails forward and back, and it can also across the area, and it can also adjust the height of this imaging box according to the height of the vegetation. What the researchers did here, and also published in the paper, is they had different levels of nitrogen, from low nitrogen to high nitrogen fertilization, and they had different wheat cultivars that were exposed to this different fertilizer supply, and they monitored then the effect of cultivars versus the effect of fertilization, not only by that NDVI, but in the paper there's much more information also about different types of imaging that they applied with the system. What you can see here that, for instance, two cultivars at low fertilization strongly differ in NDVI. This cultivar Crusoe, for instance, is obviously less fertilizer demanding. It has much more vegetation, a higher NDVI, less gaps, low NDVI in between, compared to the other cultivar, which has lower values in NDVI and much more gaps. And when we follow that up, at higher fertilization, there are still little gaps in the vegetation. So the plants grow, but they are not yet optimally fertilized. And at the even higher and highest fertilization levels, they grow then even better, and the vegetation is much more dense, because we can see here from an area where the NDVI is more or less everywhere close to 1. Finally, at over-fertilization here, the plants are again growing worse. So that is also an effect that you might be able to see when you overdose something. Then you can also detect it in the phenotypic outcome that the plants are no more increasing in performance, but they can decrease. So there are different types of information that you can derive from such scans, from such image acquisition and image processing tasks. This is a different example. it's more or less a demonstration experiment. We did it in our lab with hyperspectral cameras, and these hyperspectral cameras cover the range, the one camera between 370 and nearly 1000 nanometers. It's a visible to near infrared scanning camera, and the other is the shortwave infrared camera from around 900 to 2500 nanometers. And we put two types of leaf below the camera. There is a leaf that has a normal wax cover, and that's a scanning micrograph. That is not with our camera. That was done in a different lab just to visualize how the surface looks like. The surface has wax, and you see these crystals here, and the crystals make the leaf hydrophobic, so no water stays on the leaf when we spray it. There is a mutant that has less wax. There are some crystals, but there are big gaps. And when we spray water, the water stays there because the leaf is not hydrophobic. From a farmer's point of view, you never would cultivate this because when it rains, all the leaves become heavy and the plants will probably fall down, bend down and start molding. So farmers are, of course, happy when it is like that. But having that mutant, we were able to use it as a demonstration case because we know the surface is different. The chlorophyll and all the biochemical properties, they obviously don't differ too much because the plants are growing, they look green. And when we then look in the visible range, we see here in blue and red, both lines, both genotypes are absorbing. There's a little difference, but not too much. But when it comes to the near infrared and shortwave infrared ranges, we see that there is a difference in response of the low wax compared to the normal wax mutant, or the low wax is the mutant, the normal wax is of course the wild type. And it pretty much demonstrates that these ranges of the spectrum are responsive to surface properties of the samples. So then also a question, where do I measure? Where do I expect the differences? And of course, it's always clever before you start measuring to select the suitable tools, because when we are looking for internal pigments, plant pigments that absorb light, this is the right range of wavelengths. When we are looking at outer properties like the surface, like cell wall and epidermis, then those ranges might be better to look at. In the end, frequently the goal is to link spectral properties with biochemical traits. The classical approach is that you record data sets with cameras, but also with biochemical tools. So some lab samples of these plants, and then connect them, for instance, in a machine learning process, and set up a classification procedure out of both data sets. So in the end, when you know which wavelength range is important for a certain biochemical property, you can use the wavelengths as an indicator for your biochemistry. So that means when you want to screen something, you can use the spectral imaging to go through a big amount of samples, and only those where you see some interesting differences between, let's say, the reference and the testing group, that it is an indicator that's worth looking at the biochemistry. So you don't need to put all of them into the lab, in the biochemical lab, when you can sort out the non-interesting already via an imaging procedure as an application example. Other application examples come also from plant breeding. And here's a customer example from the Nebraska University, which has one of our systems running with a hyperspectral camera inside. they are breeding for forest trees that better cope with climate change, and climate change frequently makes drought, as we have seen also here in Europe in the recent weeks and months. There was much less rain compared to normal years, and of course also the forests are experiencing that. So it's a very important task to breed for trees that can better withstand drought. And for trees, of course, it's not only important that the leaves are proper growing, but it is also important to look at the stem. In this case, they were able to use this hyperspectral data set to differentiate between leaf and stem, because NDVI was indicated for the leaves, while other ratios here out of different wavelengths were indicated for the stems. So they could separately assess how the stems and how the leaves are performing and use that as an information input for the breeding process. As said, there are much more indices beyond the NDVI, for instance, this Vogelmann index or Marchioni index, but there is a long list of potentially usable indices that can be assessed with hyperspectral or multispectral imaging. A further example that comes from plant pathology, and plant pathology, and that was a project where we, as the company LemnaTec, worked together with some universities in Germany. We got some funding from the Agriculture Ministry, and we were looking for a method to use spectral data for discriminating between healthy and diseased tissue, as it's also related to some student work in that project. Excuse me that there are some German words in between. It is here the vasculature, which is the midrib of the leaf. There is this healthy tissue, which is marked in green. And these lines here correspond to the spectrum. So you see there is a different response of the normal tissue and the vasculature in the spectral response, in the spectral signature. You see here this absorbing by chlorophylls. There is some absorbing by flavonoids. And when there is a disease symptom, and inside the disease symptom there are phenolics and other substances formed, these take an influence on the spectral response. So in the end, we could set up an indicator of disease that not only works when the disease is already visible, but it also can see disease responses before a human can see visible symptoms. So it is a bit of pre-symptomatic diagnostics, and in this case, work was done with an expert group for machine learning that were working at the Technical University in Darmstadt, who set up a procedure to discriminate between the different responses and situations of the plant disease process. I talked a lot about imaging and image processing, and of course, when we develop systems for customers, we have a software package that does the whole process, starting with taking images, so the control of the cameras and the lights and all other hardware parts if required, that stores and handles the recorded images and data, that also gives the capability to process images, and it includes tools for machine learning, and which is a specialty of the system, is there is an option for user programmable image processing with LemnaGrid. Usually when you have images, you need to process them, you need a software specialist who is skilled in image processing. In our case, the software specialists have set up a library of frequently used functions with filters and image processing tools that are represented as such boxes that can be combined on a graphical surface. Of course, you still need to learn how that works, how you combine those functions. And as a biologist, I have to say that's a learning process that takes some time. So it's nothing which really works plug and play. You just open it and you're able to do magic. But with some learning, also as a biologist, as a lab technician, without programming skills, you are able to do some steps to set up something for image processing or to modify something that the software specialists have preset with the system. So you have some flexibility that you can set up image processing or modify existing image processing without the demand for a software engineer who can do command line programming. In the end, when images are processed, of course, the software package also contains functions for exporting the data and for visualizing them. By the way, exporting means that you can link up with any statistical software or databases, so you can work with the processed data as you usually also work with other outputs of measurement systems. So how can it look like? We have already seen such an image box. This was an example of a machine out of our PhenoAIxpert series, the so-called PhenoAIxpert Pro, meaning that you have two cameras looking at the sample. The simple PhenoAIxpert only would have a camera looking down. And yeah, here in this case, it is with the multispectral illumination. There's coming soon a PhenoAIxpert HSI, that means that is for hyperspectral imaging. There are other instruments, the HyperAIxpert system, that is, this machine has an exchanger for different imaging and sensing equipment, so that automatically images samples that you can put here on that area. So you have different imaging options and they are automatically exchanged. There are also larger systems like XYZ moving systems in this so-called PhenoAIxpert that goes across an area of potted plants. Or you have already seen that field scannalizer model that is standing at Rothamsted, where I have this exemplary data. And there are also inbuilt systems for greenhouses where plants are automatically transported to imaging stations like this PhenoAIxpert HT or HTC. The difference between HT and HTC is the one is with the C is a compact thing, while the HT is a large thing that fills the complete building, and plants are fully automatically transported into the imaging area. Yeah, with that, I had a little insight in the different technology options that we can combine with hyperspectral and multispectral phenotyping. Also a little insight in the software that is related. So with that, I'm at the end of the webinar.